Thanks. It's really been a pleasure to be here. As I told several of you, we have a 16-year-old who is interested in engineering programs, and we did a little tour of engineering schools, and we looked up engineering programs on the web, and Georgia Tech always came up on top on all these. It's an amazing place. So next time we'll visit Georgia Tech. We didn't make it quite that far south, but it's really an impressive place. You guys are lucky to really be able to call this your home. The little bit I saw is really impressive. So, all right. So today I'd like to tell you about some relatively recent work in the laboratory, and maybe it's a little bit too dark. My slides are not. Okay. Good. All right. And so. In the laboratory. And really our work is focused on mammalian neural circuits, focused on the cortex. And as many know, cortical circuits are cortex is parcellated into multiple functional regions. Excuse me, one more request. Maybe you could just turn off the lights behind the slides just to enhance the contrast, if possible. Wonderful. That's good. So cortex is parcellated into distinct areas that have distinct functions. And if you kind of drill into cortical circuits, you have complexity, cell types that are connected in highly specific circuits. And then, of course, you have complexity also on a mesoscale. Cortical regions are connected to each other to form multi-regional circuits. And as you know, action potentials represent information in the brain. Ensembles of neurons that fire action potentials represent information in the brain. And so we'd like to really understand in the laboratories how these ensembles of neurons, these action potentials and ensembles of neurons really represent information. What drives these representations of information on the one hand, and then how does the animal actually use these representations for behavior? And we address both of these questions in behaving mice. And more specifically, we study circuit computations in the somatosensory cortex. And as you've heard from Garrett Stanley's laboratory's work, for example, mainly in the context of whisker-based somatosensation. And somatosensory cortex is really all about fast time scales, we think. Millisecond time scale computation, essentially signal processing from the sensory periphery. And over the last 5 years, and this will be the focus of my talk, we've really also looked at frontal cortical areas, in particular an area called ALM that more implements slow time scales related to, loosely speaking, deliberation and short-term memory, motor planning, things that happen over time scales of seconds. And one of the things that I'm really interested in, and something that we'd like to figure out over the next 5 years, how with very similar kind of hardware, similar kinds of neurons, similar kind of biophysics, do you get actually slow time scales in these frontal cortical areas and these very fast time scales in sensory areas. And that's something that at the very end, I'll show you some stuff hot off the press that might have some bearing on that, okay? So we'll talk really mostly about motor planning, short-term memory today in frontal cortical areas. So let me start by explaining what motor planning is and why you should care about it, okay? So as you know, many movements are really too rapid, right, to really be amenable to online corrections. And these movements, as psychophysical experiments have shown, are in some sense pre-programmed. And really evidence for such a planning or motor planning process comes from behavioral experiments. So when you give a subject time to plan movements, the subsequent movements, for example, a baseball pitch, are faster and more accurate. And people have shown this. This goes back to the 70s and 80s, mainly human psychophysics, and it's also been done in non-human primates to some extent. So motor planning is therefore an important aspect of motor control, and it can precede the movement by many seconds, right? For example, during the planning and wind up to a baseball pitch. Now, second, equally relevant to this talk, is that motor planning is also a short-term memory. You could think of it as a prospective short-term memory that links past events and future movements. So in this picture, you have a pitcher that plants a pitch instructed by essentially sensory information from the catcher. The hand signals catchers, and the hand signals of the catcher, and many seconds, of course, elapse between the instruction and the movement. So the instruction and motor plans are presumably stored over this time scales of seconds in the pitcher's brain, somewhere neural representations in this brain, bridging the time from the instruction, from the hand signals, to the movement, here, the pitch. All right, now let's go to neurobiology, right? So what is really motor planning in the context of the brain? Well, in the lab, motor planning has been studied in the context of so-called delayed response tasks, okay? Tasks in which a sensory stimulus instructs an action, but the action is only executed after some memory period, or in the technical jargon of the field, a delay epoch. Okay, it's shown like here. So here, this is a really famous experiment. It's really one of the first cognitive neuroscience experiments, actually one of my favorite experiments in not just this field, but in neuroscience in general. This is from Everett's lab from the 70s. And what they did is they instructed a monkey to either push or pull a lever, but the monkey was supposed to execute the push or pull movement only after delay epoch lasting seconds. And you can see something curious here. And you can see preparatory or activity that is now referred to as preparatory activity, which is thought to be the neural correlate of motor planning. This neuron seems to anticipate the push movement, right? Instructed to push, pushes right here. This neuron, these are action potentials or spikes. The spike rate is elevated long before the movement, anticipating movement. This neuron actually shuts off during the movement. Okay, so neuron that anticipates a particular kind of movement. This is specific to the movement because when the monkey is instructed to pull and he pulls, this neuron is actually silenced. Okay, so anticipating specific neuron movement, and it's movements that the neuron cares about rather than instructions, because when the monkey is instructed to pull but he pushes, spike rate is elevated. So it's related to the future movement, okay, this activity, rather than the past instruction. So preparatory activity, this neural correlate of motor planning, right, is really activity that anticipates future movements, often seconds or longer before the movement. Okay? Now, in the brain, as you know, short-term memories in general are represented by changes in spike rates that can be maintained in the absence of sustained input over time scales of seconds. And preparatory activity during the delay epoch is an example of such a memory trace, right? Here's the instruction, preparatory activity is maintained. This is an example of a cellular memory trace. And so for these reasons, really the mechanisms underlying motor planning and preparatory activity, I think, are of broad significance in brain research and have received a lot of attention over the last, really now 40 years or so. So the kinds of questions that we want to ask in rodents, where we can do more comprehensive analysis of the phenomenon, more mechanistic analysis, how is preparatory activity organized spatially and temporally? Is preparatory activity here, this stuff, is this actually causally related to future movements, right? Or is it just some epiphenomenon? And perhaps most important for the purposes of my talk here, how do neural circuits maintain preparatory activity? And here the mystery is that neurons have time constants of milliseconds. They're essentially memoryless, but the circuits here maintain memory of a stimulus or motor plan over time scales of seconds, even tens of seconds. This is an emergent phenomenon of cortical circuits. How does that happen? Okay, and by studying motor planning in mice, we can begin to address these kinds of questions. So we started by developing a delayed response task for mice. This is almost identical to the kinds of delayed response tasks that have been studied in non-human primates for 20, 30 years or so. Here, mice perform a sensory discrimination, either an auditory or tactile discrimination. I won't talk at all about decision-making, all that kind of stuff. It's interesting in its own right. But they signal their decision about the sensory stimulus by either licking left or right to a target. Okay? But they only make the decision known by licking left or right to a target after delay epoch that is imposed by the experiment that can last seconds. This task is now pretty widely used in the field. And this is how it looks in a recording apparatus. So here you have a mouse, okay, performing the task. This is actually a tactile version of the task. Again, the details with sensory discrimination that don't really matter. The pole comes within reach in one of locations. The mice touch the pole with its whiskers. And then dutifully, during the sample epoch, the mouse holds its tongue before signaling its decision with skilled directional licking. And these are individual licks here. These are trials right in the vertical dimension, time in the horizontal dimension. And so as behavioral output, okay, we measure this skilled directional licking. And throughout the talk, blue refers to rightward movements, red to leftward movements. Okay. And behavioral experiments, like in previous experiments from non-human primates, show that mice actually use the delay epoch to plan precise and accurate movements. This is just shown in one experiment here, that if we allow the mouse time to plan movements by lengthening the delay epoch, the subsequent movements become faster. The reaction time becomes shorter, and also the movements become more accurate. So this task is robust and quantitative and really provides a tool to dissect the neural mechanisms underlying motor planning, short-term memory now in mice. All right, let's go to neurons now in mice. Okay, so as in the Tanji and Everett's experiments, memory-related neural signals is now a neuron recorded from the frontal part of the cortex called anterior lateral motor cortex. I'll tell you a lot more about that. That shows preparatory, bona fide preparatory activity. So here now you have a raster plot. I think all of you have seen raster plots. These are now trials arrayed in the vertical dimension, time in the horizontal dimension. Each circle here corresponds to an action potential. Okay, rightward trials, leftward trials. This is when the animal moves. This is the memory period. This is when the instruction, decision-making, so to speak, happens. And these are the spike rate that corresponds to these rasters. And I'll show you a lot about of these spike rates and relatively few of these rasters. Okay, so the spike rates are extracted as simply just the rate of action potentials averaged across trials, okay, for the purposes of this talk. So you can see that this neuron starts anticipating rightward movement, becomes selective for rightward movements, maintains the selectivity during the delay epoch, and then shuts off in the peri-movement period. Importantly, as required for preparatory activity on error trials, when the animal is instructed to move left but actually moves right, the neuron becomes again selective. So this is a neuron that anticipates specific movements rather than caring about the past instruction. So this tells us really that we've developed a mouse system to study motor planning and short-term memory. So where can we take this? So as I already said, motor planning and preparatory activity have been studied for nearly 40 years in non-human primates. And I think what our studies in mice have provided is a complementary set of mechanistic insights. And I'll give you some of the highlights of the last, we've been working on this for about 4 or 5 years. I'll give you 4 years, maybe 5 years. I'll give you some of the highlights, some published stuff interspersed with unpublished stuff, together with some things that are very much in very much early stages. Okay, so we started with localizing preparatory activity and motor planning. And I think this is really very important because I think localization is absolutely critical of both a behavior, essentially kind of a computation, and a particular activity pattern for mechanistic analysis of these things. You need to know where it is in the brain or how widely it is distributed. Okay, and so we've localized motor planning and preparatory activity to a small part of the motor cortex referred to as anterior lateral motor cortex. Okay, and so how do we do this? Well, the brain is a big place, and so what we started off is by doing a loss of function experiment. So this is a mouse brain. It contains about 100 million neurons. You know, we think on the order of 60 or so brain areas. We wanted to ask which brain regions are critical for planning a movement, okay, so which brain regions are critical during the delay epoch for the kind of behavior that I've shown you. And with optogenetics, we can now do this in a spatially and temporally precise manner. And just one slide about methods, because this method we use over and over again, okay, so it's going to be important. It's something called photoinhibition. It's our way to inactivate small chunks of cortex. We put channelrhodopsin into GABAergic neurons. In transgenic mice, this is relatively straightforward. Some of you know this very well. Then we can shine light onto small chunks of tissue, like so. Drive GABAergic neurons. GABAergic neurons all are local, okay? And it's really the excitatory neurons that carry information out of a chunk of tissue. And these excitatory neurons are silenced when these GABAergic neurons, or inhibitory neurons, are driven. So we can, with timescales of tens of milliseconds, with time precisions of tens of milliseconds, take away about millimeter scale chunks of tissue corresponding to individual brain areas in a highly, in a reproducible and reversible manner. So these are the inactivation methods that we use. And it turns out that these transient local perturbations are also very informative and a powerful tool to probe circuit mechanisms, which shall become important later. So we do this in a comprehensive way. We inactivate one of 55 cortical regions spanning most of the usual suspects that might be involved in motor planning, including all parts of the motor cortex, somatosensory cortex, and the parietal cortex and other brain regions, one brain region at a time, and then look for, when we silence that chunk of cortex during the delay epoch, silencing which brain region might have an effect on future movements during the peri-movement period. And it turns out that the experiment, the result is relatively simple. So it turns out that inactivating here the magenta region in this plot during the delay epoch subsequently causes an ipsiversive bias in movements, and silencing any other cortical region, singly or in combination, has no effect. And this really is a functional definition of ALM, anterolateral motor cortex. Sorry, I'm supposed to stand here and point there, so apologies over there. This is essentially a functional definition of ALM, anterolateral motor cortex, as the brain region, at least in this task, critical for motor planning. Okay? So now, how does this now relate to preparatory activity, the neural correlate of motor planning? So we performed an experiment where we mapped neural activity now in the motor cortex and also other regions with electrophysiology and imaging to ask how widely is, excuse me, I'm going to quickly, should probably, I'm going to kill the Wi-Fi. Apologies. So how widely distributed is preparatory activity? How does it compare to the functional definition of ALM in the context of motor planning? So we can do now with new genetically encoded probes, you can do new microscopes, you can do large-scale imaging, you can image large numbers of neurons simultaneously, extract activity patterns corresponding to individual neurons, and then correlate these activity patterns to behavior, for example. You can do similar things with extracellular electrodes. Doesn't make as nice of a movie. And then I'll give you just one piece of data from the imaging, and the electrophysiology is very similar. So here we took that kind of a data and now plotted in terms of what the neurons code, whether or not they're selective for movements or selective for the sensory stimulus, and how they're spatially distributed here across the motor cortex. And this is time in the trial, okay? Movement onset is at 0. And you can see this during perceptual decision making, this flow of information from sensory information from the more posterior areas to more rostral areas, and then the emergence of movement selective activity here in this anterolateral area, mainly in the deep layers. I'm going to stop this right here. So the earliest onset of movement-related activity before the movement, that is preparatory activity, is really limited to a very similar region that overlaps the anterolateral motor cortex. So there's this parsimonious convergence here of ALM as being the place where preparatory activity is expressed, and also a region where lots of function experiments perturb future movements. And I think identifying, as I already mentioned, the key brain regions is a critical step in mechanistic analysis. So now let's perform a detailed analysis of preparatory activity in ALM. I'll just show you one way we think about preparatory activity there. So on the left is the neuron that you've seen earlier. On the right are 4 additional neurons. These are peristimulus time histograms of 4 neurons to just highlight kind of the diversity of stuff that you might see on one electrode, intermingled neurons. So there are neurons that are selective for rightward movements or leftward movements, and they come in about equal proportions on each side. So that's why here in this schematic we've got blue triangles, red triangles corresponding to rightward and leftward selective neurons, and they're kind of salt and pepper here in the ALM. Moreover, you have neurons that are selective early, right, that become selective during the sample epoch or instruction epoch when the sensory stimulus is there. Others that maintain selectivity during the delay epoch, yet others that seem to become selective during the delay epoch, yet other neurons that are most selective in the peri-movement period, perhaps consistent with driving even a motor command in downstream motor centers. Now, of course, mice use the whole neural population for behavior, not individual neurons, right? So how do we make sense of this complexity? So, and it turns out the dimensionality reduction methods really provide a remarkably simple picture of preparatory activity, especially during the delay epoch and the relationship to behavior. In particular, during the delay epoch, most neurons either ramp down to a movement or ramp up to a movement, and we'll focus now on memory-related activity during the delay epoch. We record typically 30 neurons in ALM simultaneously with 2 shanks of a silicon electrode. This is now extracellular recordings. Okay, for most of what I say from now on is extracellular recordings. Imaging won't make another appearance. And you can think of activity of 30 neurons essentially corresponding to a 30-dimensional space where each dimension in this space corresponds to the firing rate of 1 neuron. And then during behavior, activity patterns correspond to trajectories in this 30-dimensional space. And the trajectories for rightward movements and leftward movements are different on average, right? Because different neurons have different firing patterns on average in this activity space. And so we can define a coding direction as the direction that best separates, okay, dynamics in this multidimensional space for rightward movements and leftward movements. And we refer to this as the coding direction. It's a simple discriminant analysis for the aficionados here. And then we can project the activity to this coding direction that's shown right here. Okay, and in this projection, this is by construction, the trajectories separate nicely during the delay epoch here up to a movement, okay? And more importantly, activity in this direction actually accounts for nearly 50% of the variance. And it turns out it's all of the variance that is relevant at least to the direction of movement and to the timing of movement, we think. So the activity along the coding direction explains behavior. Okay, let me show that to you with an analysis of single trials. Okay, so here in the schematic now we have these average activity trajectories in the coding direction, okay, up to end of the delay or movement onset. Now the mouse, of course, has to use single trials for behavior, right? And if there's a relationship between activity, these activity trajectories in this coding direction and behavior, we expect a relationship between single trial trajectories and behavior. And the single trial trajectories vary from trial to trial. So for example, if the trajectory location close to the onset of movement might relate to the direction of movement. So here is a trajectory that's close to the average right trajectory, a little bit beyond. Well, we might expect that this corresponds to rightward movements with high probability. For trajectories that lead towards left trajectories, for right trajectories that move towards left trajectories, are closer to left trajectories, maybe we see leftward movement with some probability, and so on and so forth. And conversely for left trajectories, right? And this is exactly what we see in the data. So if the trajectories in a single trial are closer to the average right trajectory, we get correct behavior, rightward movements with high probability. Conversely, for leftward trials, and as the trajectories leak to the trajectories for the other movements, errors creep in. So that's pretty good evidence that ALM activity actually is related to future movement, that this is really causally related to future movement. This is really a key area for motor planning. But there's still some possibilities that this area where we did this analysis might inherit these fluctuations from somewhere else, right? And might just sort of, and might not actually play causally a role in movement. So we thought, why don't we just introduce perturbations and fluctuations directly into the ALM network, and then do the same kind of trial-by-trial analysis. And we've done this experiment about half a dozen ways, and the take-home message is always the same. I'll just give you one experiment, which I think is particularly illuminating. Here, we transiently inactivate in such a manner that we completely obliterate, okay, preparatory activity on average. Okay, so the trajectories on single trials, this is just illustrated here for some right trajectories, lose all memories about what the trial type was, and now meander randomly subsequently, okay, in the coding direction, and might equally likely end up here closer to the rightward movement trajectory or the leftward movement trajectory. Yet on a trial-by-trial manner, we can still read out movement direction with pretty good reliability. So by analyzing the perturbations of the effects of perturbation both on neural dynamics and behavior, we can show that I think preparatory activity in ALM in fact instructs future movements. All right, so let's now go to a more mechanistic analysis about maintenance of preparatory activity. So how is preparatory activity generated and maintained in actual neural circuits? And we don't have all of the answers. I'll just give you sort of an update of where we are, what we've done, and how we're thinking about the problem. So we have discovered, really by accident, mechanisms underlying robustness in maintenance of preparatory activity. And that's what I'd like to show you first. And so we discovered that after large-scale inactivation or activation of ALM, and I mean we can really silence one hemisphere of ALM transiently, that preparatory activity or activate ALM with channel-Robson or inactivate ALM with channel-Robson, that activity and selectivity recover in detail over timescales of hundreds of milliseconds. And that's an interesting experiment. Seems a little crude, but it's an interesting experiment because it really provides real challenges for circuit models of preparatory activity in short-term memory. So let me show you how this looks in detail. So here is one trusty neuron here. Again, neuron that's rightward selective, so it ramps up to, it's persistently active here over timescales of a second or more. And here is the inactivation experiment. We really silence it and all of its neighbors, okay, entire ALM on one side. Activity levels go to 0, as they should, because we silence. After offset of silencing, activity recovers over timescales of 100 milliseconds as advertised, but not just activity recovers, but selectivity recovers, right? On rightward movement, the neuron becomes as active as it would have been in the absence of perturbation, and the same on leftward movements. So somehow this neuron and its neighbors maintain a memory of what its activity should be despite the massive disruption of network activity. And this behavior holds at the level of populations. This is, again, the kind of dimensionality reduction method that I've shown you earlier. Now, a simultaneous recorded population along the coding direction. Here, during the perturbation, the selectivity completely collapses, but then recovers over timescales of 100 milliseconds. And with this kind of perturbation, also the behavior recovers. So how does that work? Well, it turns out, not surprisingly, that the memory has to sit somewhere else, right? Another area has to communicate with the perturbed area and help the perturbed area recover. And we did a search for these areas, and it turns out it's the other side of ALM. So here is ALM on one side, ALM on the other side, and the two sides of the motor cortex, ALM in particular, are incredibly strongly connected through the corpus callosum. This is an anatomy experiment. So a tracer was injected right here, and the axons project to the other side of the brain, kind of homologous regions on the two sides are strongly interconnected. So this is a candidate area that might help maintain motor preparation in the during the perturbations. And it turns out, as I've shown you earlier, that preparatory activity for rightward and leftward movements is on one side, and it's also on the other side. Okay, so when you perturb this side, it could be that activity recovers through the other side, and that's in fact what happens, okay? Because if we now perturb both sides of ALM, the selectivity fails to recover. So the key principles that underlie robustness of preparatory activity are really redundancy and modularity. Okay, that's sort of perhaps the more important point here. The circuit consists of at least two modules. In this case, a module in the left hemisphere and in the right hemisphere. These are coupled so that when one module is perturbed, the other module helps the perturbed module recover. And we refer to these as modules because each module can maintain preparatory activity independently when the other module is perturbed. Okay, so modularity because each module can maintain preparatory activity independently, and redundancy because preparatory activity is distributed across these two independent modules. And as engineers, you should probably know that robustness in engineered systems, right, is often implemented through redundancy and modularity and feedback connections. Actually, an important design principle underlying the Apollo mission, I hear, but it's a separate story. All right, so now each module can maintain persistent preparatory activity, and we're currently exploring the mechanisms of how preparatory activity is maintained in individual modules. And this is ongoing work. Let me show that to you briefly. Here's the problem, right? You've got neurons in one side of ALM with different selectivities. If you inject information into neuron, briefly here, different kinds of inputs, say during the instruction epoch in our behavior, the information dissipates over timescales of milliseconds, really limited by the membrane time constant, but actually quite a bit faster because of active conductances, right? So neurons are essentially, in isolation, unconnected neurons are essentially memoryless. In contrast, preparatory activity can last for many seconds, okay? So now, theorists have been aware of this problem since the days of Robinson in the eye movement field, at least 30 years, and have come up with many, many models of how neurons might maintain selective, persistent activity over timescales of seconds for the purposes of all kinds of short-term memories. And they really fall into 4 or 5 classes, some are a little bit more exotic. I've just shown 3 kind of network models in a cartoon fashion. You could imagine that neurons are arranged in a sequential chain. and hold memory in that way, where one set of neurons excite another set of neurons, excites another set of neurons, and so on and so forth. Mark Goldman has a nice model about that. And we think the dynamics of our neurons is not consistent, and their response to perturbations is not consistent with this model. Then there are two perhaps more plausible models. They're both in the realm of feedback-dominated neural networks that are dominated by excitatory networks, excitatory connections. And here, in both cases, in the integrator and the discrete attractor model, the principles are similar. The time constant in the neural networks are prolonged by feedback excitation. So there's dissipation of excitation through the membrane, but then you get feedback from a neuron that has similar tuning to give you that kickback, to keep you in that excited state. And integrators are distinguished from attractors because they have a continuous set of stable state, and they respond in a graded manner to graded input, okay? Whereas discrete attractors convert essentially continuous transient input into sustained discrete outputs, here perhaps corresponding to the two movement directions. And it turns out, I won't go through all of the experiments, that really our experiments point to this model, the discrete attractor model, as underlying the dynamics of ALM. Let me show you two pieces of evidence. First, the discrete attractor model really predicts a funneling of activity over time during the delay epoch, right? So you've got some input. Sometimes the input is lower, sometimes higher. There's noise in the system, right? And the activity trajectories should converge to discrete endpoints, also attractors or fixed points during the delay epoch, okay? And that's in fact what we see in the data. These activity trajectories average in a particular way. I can't tell you about the details, but they seem to converge to discrete separate endpoint during the delay epoch, and this has been noted by others, okay? Perhaps, and there are several explanations for this. Not all of them require this to be a discrete attractor. Perhaps a more incisive and stringent test, again, comes from these perturbation experiments, okay? And this experiment I like a lot, okay? I'll go through this in some detail. So what you expect for these discrete attractors is if you push the system far, but not far enough, not across essentially the point that separates the two attractors or endpoints or fixed points, but also referred to as a separatrix in the language of dynamical systems. Some of you may have taken a course in that. The attractor should recover to its normal endpoint. If you push, on the other hand, the attractor across the network, across the separatrix, you should recover to the other attractor, of course, with an error now in behavior, right? And so, and this is what we did. So this is, and when I say we, this is really the ingenious work of Hidehiko Inagaki, who did this experiment despite my advice. I didn't think this would work for various reasons. As usual, I was wrong, but anyways, that you know all of that stuff too, yeah. So anyways, so here are the trajectories under control conditions. He devised a perturbation method that collapses the selectivity, has relatively little effect on mean firing rate. This is sort of a subtle way on some subtle point on some trials. The perturbation, the correct trajectory is recovered. That produces correct behavior. On other trials, the trajectories flip. Okay, rightward trajectory now snaps into the leftward trajectory, and vice versa with an error trial, exactly as predicted for discrete attractor models. And I think this is the strongest experiment that we have that really points that discrete attractor dynamics underlies preparatory activity. And we have an idea of how the basic circuit motif looks like based on analysis of connectivity. We think that neurons with similar selectivity and dynamics, neurons that are early active, late active, connect to each other preferentially and recruit inhibition in a relatively fast spiking into inhibition in a relatively promiscuous manner. And it turns out that models of this type of the circuit motif actually produce attractor dynamics without too much fine-tuning of the parameters. I'm going to skip this. It's just some more details about the model and tell you a little bit about now more localization. I'm going to go through this very quickly because I wanted to show you the last bit, which is just really more far out. And so I'm going to return now very quickly to the topic of localization, now in the context of multi-regional circuits. So far I had this myopic view of one brain region. And it turns out that's by far not the whole story. And it turns out that, and we were interested in this because, again, it's a question about mechanisms, right? Most models of preparatory activity, short-term memory, really rely on excitatory connections through cortical horizontal connections, so local. This is a cortical neuron. Cortical neurons make a lot of axons locally, and people think, well, it's these connections, right, within ALM, for example, that maintain memories. And multiple lines of evidence from our lab indicate that a cortico-thalamocortical loop plays a critical role in maintaining persistent activity. And the experiment is very simple. We looked at other areas that are strongly connected in a bidirectional manner with ALM, and asked which one of these might be critical for maintaining preparatory activity. And it turns out that the most strongly connected areas, the cortical areas, have no effect. And I've already shown you this from the other side of ALM, right? You can blow out one side of ALM, preparatory activity in the other side is maintained, similar for primary motor cortex and similar to sensory cortex. In contrast, if you silence thalamus that projects to ALM, you have this absolutely stunning effect on ALM activity and selectivity. Completely, cortex becomes comatose, basically. And the converse is also true. If you silence ALM and record from thalamus that receives input from ALM, selectivity and activity is lost in thalamus. So we think that each side of this module on each side that maintains preparatory activity really relies on feedback between a cortico-thalamocortical loop. Okay, that's required to maintain activity and selectivity. I haven't shown you all the pieces of data. This was actually published a couple months ago. All right, let me briefly, very, very briefly summarize. So we've identified the key neural circuits underlying motor preparation. During motor preparation, preparatory activity converges to endpoints. And actually, the Chenoy lab refers to this essentially an initial condition for movement in activity space. And I think that's probably the right picture. Reaching the appropriate initial condition is critical to produce fast and accurate movements. And I think these manipulation experiments show this. And preparatory activity is these fixed points correspond to these initial conditions correspond to discrete attractors and preparatory activities implemented in modular and redundant circuits. All right, so now so far our models of preparatory activity are algorithmic in nature, right? So they're basically to a first order embodiment of hypothesis of how the dynamics might be generated, right? These kinds of, and it turns out that these kinds of network motifs that are modeled are relatively difficult to implement with actually biologically plausible neurons. And I give you just one example here. This is a cartoon that I made this morning, so it's a little rough. But the problem is one of inhibition and time scales of feedback, all right? So if you have a neuron that spikes and then evokes activity in inhibitory population, the feedback inhibition comes in very quickly before the next excitatory spike is fired, okay? And that shuts off activity in these networks. And so modelers have a workaround, and so what they do basically is they say, well, look, let's just make synaptic time constants 100 milliseconds. Now, I don't know if these synaptic time constants, but that bridges, right, these these time scales of feedback inhibition. So there are these kludges that are sort of hidden in the methods. So I think that the biophysics really matters, okay? And cell types really matter. And this is something that, of course, we can take seriously now, because this is really the era of cell types. And I think, and so we, in a collaboration with the Allen Brain Institute, have done for the last few years, and they did all of the heavy lifting with respect to single cell sequencing, done some essentially classification of cell types in the anterior lateral motor cortex. And what we find is, I'm going to focus only on 2 clusters. This is clustering of we think there's about 40 neuron types, so extremely complicated. We have a lot of work to do. This is a lot of the stuff going on in the lab is informed by this data. This is now clustering by gene expression, right, by transcriptomics. I'm going to just focus on 2 clusters here in ALM. Down here, this corresponds to the major output neurons of ALM in layer 5, because I think they are critical for maintaining preparatory activity and movement initiation. They're relevant to the story of maintaining persistent activity. These 2 clusters correspond to different projection classes, okay? And this is done by combining gene expression data with structural analysis. And this is a data set from Mouse Light, a shared project at Janelia. Mike Econo was one of the key players, a postdoc in the lab. And these are single neuron reconstructions, 4 neurons overlaid. Cell bodies are here. And they fall into 2 classes. There are neurons here, the yellow and green project to the thalamus. The magenta and blue project to the medulla, to premotor centers that also control orofacial movements. That's really the premotor centers that control the tongue, are critical for speaking, and so on and so forth. So 2 cell types corresponding to these 2 gene expression clusters, and they overlap in other places like the superior colliculus. Now, here they are. So one projects to the thalamus, one projects to the medulla. And what is very exciting is that the projection from the cortex to the thalamus projects to the thalamus and essentially ignores GABAergic neurons in the thalamus. Does not excite thalamic reticular nucleus neurons. And this is data from a collaborator, Gordon Shepard at Northwestern University. This is a cartoon based on data from Gordon's lab. So this could be a specialization for maintaining long time constants in frontal cortical circuits. And this is how we're thinking about the problem now. This is a thalamocortical loop that is excitatory only, avoiding this problem of rapid excitatory feedback that has a tendency to shut persistent activity off. Now, there is a second specialization, which is pretty wild. When we look through the gene expression data, we find that the thalamus projecting neurons express at high levels an exotic NMDA receptor, gluN3A. You know, I used to work on NMDA receptors. I'd never heard of this thing. There are like 5 papers on it, but it has incredibly interesting properties for the purposes of short-term memory. It has a long open time, 100 milliseconds or so, and it lacks a magnesium block. Does everyone know what a magnesium block is? Yeah, so NMDA receptors are voltage dependent. That's why they're not really that useful for maintaining this persistent activity. And they're voltage dependent because of magnesium block. These things lack the magnesium block, so they're opening at rest with glutamate binding. And also, they don't conduct calcium. So they presumably don't induce synaptic plasticity. So they're perfect in terms of dynamics and properties to maintain long time scale activity. It's in fact is exactly the properties of the kind of receptors that modelers have used for a couple of decades. So we don't know yet if they're even at synapses. This is kind of a working model that these might be critical to maintain preparatory activity. But this is something in the age of CRISPR that we can find out. But last piece of data So we hypothesize, and these guys then project to motor centers. We hypothesize that this loop is somehow critical for maintaining preparatory activity, and this kind of branch to initiate movements or giving pre-movement periods. So we did some cell type specific recordings in these two neuron types, and this appears to be the case. So what we're doing is recording in ALM and then exciting these different neuron types by backfiring them through either from the medulla or the thalamus, and then identifying neurons that project here or there in ALM. And when we now look at the persistence of selectivity in the thalamus projecting neurons, we see that they become selective remarkably early, maintain selectivity up to the peri-movement period through the delay epoch. Okay, so there's memory, short-term memory in thalamus projecting neurons, sort of as hypothesized, not in medulla projecting neurons. In medulla projecting neurons, they show very rapid changes in selectivity after the go cue, consistent with triggering movements, and these don't show that thalamus projecting neurons don't show that kind of activity. So we think that there is different output neurons in layer 5 appear to be specialized for either maintaining motor plans and initiate movements respectively. And so, of course, one of the key questions that we're interested in is now how this transformation from motor planning to movement initiation is achieved, right? This is one of the key questions about how information is gated from one brain area to the other, and we're very much working on that. In general, I think in this ALM circuit, we now have the opportunity to link neural circuits to neuronal biophysics and to neural computation and behavior, and we've sort of opened the black box, right? And we know the relevant brain regions, cell types, how some of the dynamics relate to behavior. We can measure key state variables using imaging and neurophysiology, and optogenetics allows us to shape state variables, and with methods like CRISPR, we'll be able to get at molecular mechanisms. So we have our work cut out for ourselves over the next few years. I'm going to skip all of this and just thank all of my collaborators. And let me highlight a few folks in particular. Nuo Lee and Zhenkai Guo have started this work with me, and they're incredibly cool guys. They're now off. Keivan, Hidehiko, Mike, and Sarada are carrying the torch onward. Hidehiko is on the job market. He's absolutely brilliant. Okay, this guy is also going to go on the job market. This guy also. Sarada will take a while. And then very valuable collaborators in matters of theory and data analysis, Shaul Druckmann, Sandro Romani, Lorenzo Fontolan, and other collaborators whose work I only was able to skim. And thanks for your attention. And I'll take questions if there are any. Yeah.
So I'm very curious, the dimensionality reduction that you observed in the network protein, this screen factor, if you think that that's a sort of common feature of these premotor centers or the preparatory centers for sort of all kinds of behavior, or it could be in part because the task that it's preparing is low-dimensional.
Yeah. Oh, absolutely. So this is critical, right? So in all of these tasks, the dynamics of the activity to some extent reflects the dynamics and the dimensionality of the behavior. So the dimensionality is low because the behavior is low. So and we think that attractors are learned, and that if we train additional endpoints into this, we'll get additional attractors, and this is the kind of thing that we're trying to that we're doing. Yeah. So the dimensionality will not in general be as low dimensional. So Surya Ganguly has done some analysis on various data set, including Krishna Shenoy's data set. This is a reaching task, and he's come to that conclusion. We have an intuition that might be the case, but sort of has put more quantitative meat on this notion that the dimensionality in these motor net dynamics really reflects the dimensionality of the behavior. Yeah. Yeah, so so whether or not you get discrete attractors, that's a good question. So this is something that is, so what we're doing is training in additional targets and trying to see whether or not that looks, this is ongoing, corresponds to additional discrete attractors. I think really the question is, what about short-term memories that now require require representation of a continuous variable and essentially flexible use of that information, like in a working memory task. And that's also something we're interested in. And I would imagine discrete attractor dynamics would not deal with that in a parsimonious way. So yeah, that would be an interesting point for comparison. And you can, perhaps use the same kind of, unleash the same kind of experimental arsenal to look at the underlying circuit implementation. So we haven't done that yet. Yeah.
Related question. You're talking about short-term memory in attractor dynamics. And I'm wondering if what you view being remembered is something like a choice variable, or if it's actually an action sequence. Because for sequences, attractor networks don't have brain capacity, but there are other proposals for short-term memory in transient activity. Sorry, activated series.
Yeah.
For sequences, there are proposals for short-term memory in transient activity of recurrently connected networks rather than attractor dynamics. Do you think it's a decision variable that's being encoded that's more static and low-dimensional, or do you think that there are actually motor commands that could be encoded in transient activity rather than attractor?
I don't know about the decision and stuff. I don't really know. I think I personally, I like to talk about motor preparation, and I think decision variables are probably very closely linked to motor preparation. And I don't know of a single experiment that has really deconvolved these two. This is sort of a bigger picture. I would get in a huge amount of trouble in certain departments with that kind of statement. So don't spread the word. But your bigger question is a mechanistic one, right? So you're thinking about RNNs basically, and so these are recurrent neural networks, and yeah. So we think you can train RNNs to look like discrete attractors effectively, or like integrators. And in fact, if you just impose this task, they tend to want to be integrators. So it's kind of and then others. So I think and then there are, I think the principles turn out to be very similar. Although you then have to go in and reverse engineer these things, and that can be complex in and of itself. But so depending on what you train, so if you train them just to produce two fixed points, they'll produce discrete attractors. So I think those are kind of tabula rasas in some sense, and the way the details and how you at least in our experience, and we're not as good at this as we don't understand these things as deeply as some others, that you get what you train. And so I like this kind of algorithmic approach, and then you just sort of go and sort of ask what is this dynamic not consistent with, and that's if you go from there. Yeah. So that's something we don't entirely know. So there's a question of whether or not this area is specific to orofacial movements and in the context of motor planning, and we don't know the answer to that. So there's just very little work that has gone into this kind of detail. We looked for the relevant brain regions in an unbiased manner, and it's something that someone should really do in a reaching task, for example, or in a, so, early, we've done some very preliminary stuff with kind of directional navigation where it looked like ALM did play a role, but it's too early to tell, I unfortunately don't have the answer for that. Well, I think we have to vacate the room, is that right? Or, well, thanks.